Hello friends, welcome to entranceadvisor.com. So today in this chapter 11, I'll be talking about only the first half of the chapter biotechnology, that is principles and processes. Okay, so let's start. Before starting the video, if you are new to this channel, please hit the bell icon and do subscribe to our channel. So, what is biotechnology? At its simplest, biotechnology is a technology based on biology. Biotechnology harnesses cellular and biomolecular processes to develop technologies and products that help improve our lives and the health of our planet. We have used the biological processes of the microorganisms for more than 6,000 years to make useful food products such as bread and cheese and to preserve dairy products. So, biotechnology is defined as the technological exploitation of microorganisms, plant cells, and animal cells and biological processes, including the genetic engineering, for obtaining useful materials for agriculture, industry, domestic, medical, and other uses. The use of whole plant and animal is the domain of agriculture, horticulture, and animal husbandry. Human beings have been using the biotechnology for thousands of years in the production of curd, cheese, bread, vinegar, wine, manure, etc. before having seen the microorganisms involved in the processes. It was Pasteur, Kuhne, and others who found uh, the microorganisms and produced after the First World War with the help of the microorganisms. So, biotech is helping to heal the world by harnessing the nature's own toolbox and using our own genetic makeup to heal and guidelines by research by reducing the rates of infectious disease, saving millions of children's lives, changing the odds of serious life-threatening conditions affecting millions around the world, tailoring treatments to treat individuals to minimize health risks and side effects, creating more precise tools for disease detection and combating serious illnesses and everyday threats confronting the developing world. So the biotech uses biological processes such as fermentation and harnesses biocatalysts such as enzymes, yeast, and other microbes to become microscopic manufacturing plants. Biotech is helping to fuel the world by streamlining the steps in chemical manufacturing processes by 80% or more, lowering the temperature for clearing the clothes and potentially saving $44.1 billion annually, improving the manufacturing processes efficiency to save 50% or more on operating cost, reducing use of and reliance on petrochemicals, using biochemicals or biofuels to cut greenhouse gas emissions by 52% or more, decreasing the water uses and waste generation, and tapping into the fuel potential of traditional biomass waste products. So biotech improves the crop insect resistance, enhances crop herbicide tolerance, and facilitates the use of more environmentally sustainable farming practices. Biotech is helping to feed the world by generating higher crop yields with fewer inputs, lowering the volumes of agricultural chemicals required by the crops, limiting the runoff of these products into the environment, using the biotech crops that need fewer applications of pesticides and that allow the farmers to reduce tilling the farmland, developing crops with enhanced nutrition profiles that solve vitamin and nutrient deficiencies, producing foods free of allergens and toxins such as mycotoxin, and improving food and crop oil content to help improve the cardiovascular health. So, biotechnology can be defined as the use of microorganisms, plants or animal cells, as I've told, or their components to produce products and processes useful to humans. According to European Federation of Biotechnology (EFB), biotechnology is the integration of the natural science and organisms, cells, part and thereof, and molecular analogues and products and services. The term biotechnology was coined by Carl Yerke in 1919. So, the citric acid was the first non-alcoholic product that was antibiotics were obtained from them, and the level of production has increased with the genetic improvement. Penicillin yield improved by a factor of 1000. However, there has been no change in the type of recombinant technology is used to enhance the productivity and obtain new products from the microorganisms, plants and animals. The recombinant DNA technology and genetic engineering developed in 1970s when the techniques became available to excise the desired genes from one organism and transfer the same to the another organism. The first useful commercial product of transgenic microorganisms was the synthesis of human insulin by the bacterium Escherichia coli with the help of the newly inserted human insulin gene. The proteins used by transgenic microorganisms are called recombinant proteins, and a number of them are of great medicinal value. So the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. Many processes like the IVF or in vitro fertilization leading to the test tube baby, synthesizing the gene and using it, developing a DNA vaccine or correcting a defective gene are also the parts of biotechnology. The European Federation of Technology (EFB) has given a definition of biotechnology that comprises both traditional and modern molecular biology. The definition is as follows: the integration of natural science and organism cells, parts and thereof the molecular analogues of products and services, as I've written here. Repeated only. The biotechnology, the use of biology to solve problems and make useful products. The most prominent area of biotechnology is the production of therapeutic proteins and other drugs through genetic engineering. So what is genetic engineering? I'm going to tell you this. The principles of biotechnology. The modern biotechnology is based on the two main principles. The genetic engineering, which is a technique to alter the chemistry of the genetic material DNA or RNA to introduce these into another organism and thus the change the phenotype of the host organism. Another one is adequate maintenance of the sterile conditions to support the growth of only desired microbes, eukaryotic cells in the large quantities for the manufacture of the biotechnological products like antibiotics, vaccines, enzymes, etc. Let's move on. Let's move into details. What is genetic engineering? The genetic engineering is defined as the direct manipulation of genome, DNA or RNA of an organism. It involves faster and transfer of the new genes to improve the function or the trait into the host organism and thus changes the phenotype of the host organism. So it is a biotechnology involved in the synthesis of artificial genes only, repair of genes through fusion, deletion and inversion, shifting such as transposition of genes, production of recombinant DNA, that is the combining DNA from two sources and manipulating them for the improvement in the human beings, plants, animals and microbes. An important aspect of the genetic engineering is the RDT or the recombinant biotechnology or the recombinant DNA technology, in which the desired DNA sequences from the different sources are obtained and joined together, to form a new combination of the recombinant DNA, newly formed rDNA, that gives a desired product or trait. Recombinant DNA technology is also called the gene splicing as it involves cutting and pasting of the desired DNA fragments. It is based on two important discoveries in the bacteria. First of all, that is the presence of the plasmid, what is Hess and Lederberg in 1952 in bacteria which can undergo replication alongside and independent of chromosomal DNA. That is, and the second one is the restriction endonucleases, Arbour and Nathan Smith in 1970, Nobel Prize in 1978, which can break the DNA at the specific sites. They are approximately called the chemical scaffolds or the molecular scissors. The science of recombinant technology took birth, then Berg, and was able to introduce the gene of the sp40 into the bacterium with the help of the lambdaphage. Then the Berg is often considered father of the genetic engineering, and he was awarded Nobel Prize in 1980. Cohen and Boyer in 1973 were able to introduce a piece of gene containing the foreign DNA into plasmid of Escherichia coli. So how it is done, this process? Maintenance of the sterile condition in chemical engineering process enabled for the growth of desired microbes for the manufacture of biotechnological products. Traditional hybridization using plants and animal breeding leads to the inclusion and multiplication of the undesirable genes along with the desired traits. The technique of the genetic engineering, which include the creation of recombinant DNA, use of gene cloning and gene transfer, allow us to isolate and introduce only one set or a set of desirable genes without introducing the undesirable genes into the target organism. So in a chromosome, there is a specific DNA sequence called the origin of replication or ORI site, which is responsible for the initiating replication. Therefore, for the multiplication of any alien piece of DNA in an animal or organism, it needs to be a part of the chromosome which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This is known as cloning or making multiple identical copies of any template DNA. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a negative plasmid of Salmonella typhimurium. Stanley and Cohen, as I said, and Herbert Boyer 1972 isolated the antibiotic-resistant gene by cutting out a piece of DNA from a plasmid that is an autonomously replicating circular extrachromosomal DNA of the Salmonella typhimurium. The cutting of DNA at specific locations became possible with discovery called the molecular scissors, that is the restriction enzyme. The cut piece of the DNA was linked with the plasmid DNA. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. A plasmid can be used as a vector to deliver an alien piece of DNA into an host organism. The linking of the antibiotic resistant genes with the plasmid vector became possible with the enzyme ligase, which acts on the cut DNA molecules and joins their ends. This makes a new combination of autonomously replicating DNA created in vitro and known as recombinant DNA. When this DNA is transferred into the E. coli, it could replicate using the new host DNA enzyme and make the multiple copies. Okay, the multiple copies are made from the DNA polymerase, and the ability to multiply comes to the antibiotic resistant gene in E. coli was called the cloning of the antibiotic resistant gene in E. coli. The DNA or the rDNA technology is also called the genetic engineering, deals about the production of the new combination of the genetic material or the artificially in the lab. These RDT or rDNA molecules are then introduced into the host cells where they can be propagated and multiplied further. Thanks for watching. Have a great day.